Well, I had simply to give to this is a special lesson, a special event, organized also support of I. Of course, here the course that I invite you to type in your devices. This is the code for the medicine and surgery course students, and this for Medicina and Chirurgia. Mistake. Before introducing our guest, who's, by the way, Professor Giuseppe Gasparre from the University of Bologna, I have been asked by Erich to say a few words about this initiative, about Erich itself. I guess that most of you, the Italians, in particular Italian students, will know Erich. As usual, the remote control works when I try, does not when I need it. Okay, now it works. Before delivering the lecture, please be aware of the fact that the lecture will be recorded and stored for scientific dissemination purposes by Erich, meaning that the lecture recording will be made available to the public through the Erich Campus lecture series on and so if you speak, if you speak or ask questions to the lecturer, be aware of the fact that your face and voice will be captured on camera, not easy here, but probably the voice will be, and so it will be streamed. And so in a way, by asking questions, we agree that your face and voice will be recorded. But please ask questions. We should not break you from asking questions. Is it any better at home?
Yes, now we can see them.
Very good. I don't know, Giuseppe, what you did. I think I will ask your support also for next week because I have enduring problems, as my students well know. So, what is Erich Campus? Erich Campus is an initiative of Erich, a special program designed to, let's say, bring together Erich and the Italian universities with an outlook at the future of research. For those of you who may not know what Erich is, it is a charity, the largest Italian charity for cancer research, so Italian Association for Cancer Research. And has been with us, supporting us the last 56 years. Erich was founded the year I was born. I don't know if this is a coincidence or it is. And they have been continuously supporting the progress of cancer research through fundraising, as well as especially well, with new media in the last years, they have been also involved in the dissemination of the results achieved. Just to give you an idea of the amount of money that Erich raises and makes available for cancer research. Last year, they allocated 125 million euros to fund together more than 600 research projects, 71 scholarships, and 22 special programs. At the heart of Erich are in fact several of us volunteers who contribute, as you will see, to all IARC initiatives. As I mentioned, communication is part of IARC mission. IARC and we strongly believe that knowledge is the key to beating cancer. IARC has done over the years a huge effort to make results of knowledge, also of risk factors for getting cancer, available to everybody in Italy, starting with a traditional distribution by mail of the newsletter Fondamentale, which reaches our homes, and then more recently by developing a well-structured, up-to-date website, which received more than 14 million visitors. And it has also, of course, IARC is also present in the main social networks and has a number of followers. IARC communication activities include the dissemination of the scientific results and also the awareness of projects focused on prevention, on papers, TV, radio, various websites, and 14,000 schools and 24 scientific festivals. So they try to be to show up in every good event where you have the opportunity to disseminate knowledge. Coming to our campus, that's a special project which started a couple of years ago with a special aim to reach university students. And so IARC is making some deals with the universities, including the University of Padova. Pretty sure it will be the same with the University of Bologna. Some universities have already signed the agreement, others will do it shortly. The initiative includes a series of lectures on topics related to IARC's research activity, which must be pertaining to the various courses of study. And the lecturers are professionals in various fields who acquire their specific skill through their practice and direct experience. So it's a way to enable students who, maybe some of you will eventually decide to become a cancer researcher, who knows? We were both medicine students some time ago, and the possibility to have an idea of what cancer researcher is. So the IARC community is a big one. There's in Italy 4.5 million supporters, contribute sometimes just 10 euros, 20 euros, but nevertheless, altogether is a huge amount of money for cancer research. And very important, IARC can count on 20,000 volunteers. Without the volunteers, I think IARC would not exist, or would be certainly not as is. IARC has also 140 employees, and it funded 5,500 researchers. It's organized nationwide in regional committees. So there are in Italy 17 regional committees, which are in charge also of organizing some regional activities for IARC. You can also do something for IARC, and one possibility would be that you simply join us and become a volunteer there are several ways to help iron. One is shown in the picture here. Typically, iron cells, I think this event has been celebrated for 40 years. It's a very ancient event nationwide. So we sell flowers on the Mother's Day, and we sell oranges in January. That's typically
chocolates.
A chilling experience because sometimes it snows, it rains. Like today, it's not very pleasant to be outside and wait that somebody buys 3 kilograms to an half oranges. But it's an event, and so I think that I really needs young people to contribute to these kind of events, which are not only in the main Italian cities, but also in smaller cities, sometimes in villages. So without thousands of volunteers, they would definitely not be possible. Finally, what about Veneto and IRC? The regional committee of IRC is located in Verona, and these are the contact emails and numbers. It's just also a possibility to get in touch with IRC. Could any of you also be interested in promoting IRC? So, and I think the last slide of this short institutional presentation, and we will now change and go to the lecture by Professor Giuseppe Gasparre, who is a full professor at the University of Bologna, director of the Applied Biomedical Research Center, deputy director of the Department of Medical and Surgical Sciences, and he is an IRC researcher. So he's taking, of course, since you are students, we won't present many, regional data. So the special aim of this lecture is to give you some insights and perspective into tumor metabolism. And so we are really very pleased, Giuseppe, that you are with us. And
thank you. Right, thank you all for attending. First of all, not necessarily it was going to be so with this weather and with all this possibility to follow the classes remotely, but nevertheless, thank you very much, because I very much prefer lessons with a direct, relationships with the student, and because I'm going to ask you questions and we're going to talk about stuff today, so will all be recorded at the end of the day into the IRC class. So let's start with the first question to you. Well, first of all, let me tell you what I'm going to do today. We have about half an hour. Because you are medical students, and some of you I heard are biology students, I'm just going to give you an overview. So that's the very title of the presentation is Cancer Metabolism Month, because this topic is very wide. It's extremely wide, so that there are conferences all over the world speaking on all metabolic aspects of cancer research, which is something that has come up in the past 20 years. It has boosted really in research, but it's a very old thing, because this aspect of cancer, which is very peculiar and is able to distinguish cancer cells from normal cells, comes from observations, and we'll see a little bit of a story in historical terms from the beginning of last century. So it is about 100 years that the first observations that cancer cells have a different metabolism from normal cells have been brought up. And on these basis, we have revisited them over time. Research is now going deep into these aspects, but over time, this has become really a peculiar hallmark of cancer cells. And why do we want to look for hallmarks in cancer cells? Why do we want to find cells? Why do we want to find properties that distinguish cancer cell from normal cells? Why? Yes. No one dares to speak? Come on, we're among friends. Why would you want to win the Nobel Prize by finding something that is so typical? Yes.
Like to find a therapeutic target, because if you create
Tell me an adjective for that therapeutic target. How do you want it to be? Specific. Yes. Because if you plan to use a drug,
yeah, you want to avoid side effects to the patient, so you don't want to kill the normal cells in the patient.
perfect. So this is the aim, right? So in cancer metabolism, we find a milieu of targets that can be specific for cancer cells if we know and if we learn that cancer cells use specific metabolic pathways that normal cells can do without, can live without. But it's a very broad topic, so I'm just going to give you a glance of this all. Can anyone tell me what this sketch is? Anyone seen this before? It's a drawing made by somebody who was so modest to write on top of it, I think this, but then he was so modest that he wrote a book this thick about it. That if you read a die, right after you close the last page, and you're a hero if you get to the end of it. No one? Yes.
Maybe it's Charles Darwin.
Not maybe, it is Charles Darwin. So this was the first drawing that he made about the origin of species. He thought that by starting from an organism, you could then branch out different organisms with different properties according to selective pressures of the environment. And then the organism would differentiate into different species, right? So the driver for this force is the environment, is the conditions of the surrounding environment. So this is the sketch that he drew, and this is something that we can use to model cancer. So this is the very first concept that I'd like you to. It is that cancer can be seen, first of all, as a very dynamic disease. And the cancer mass that you are maybe led to think of when it comes to cancer mass, indeed is a microreactor evolution. Within a cancer mass, you can see in an accelerated form, in an accelerated fashion, all that Darwin described into his origin of species. Within a cancer mass, why do we see the whole evolution of the species in such a short time? Because of the speed at which cells reproduce. By increasing the speed, of course, Darwin had giraffes or turtles that he was looking at, and it takes a while for turtles' generations to reproduce, but it takes less than 24 hours for cancer cells to double. So within a short time, you can see the whole course of evolution within a cancer mass. And what does this mean in terms of biology? It means that within a tissue ecosystem, there are cycles like this one that continuously repeat over time, in which you first have a genetic diversification, in which the ability of cells to reproduce implies that they change genetically. Due to what? What drives genetics diversification? Very easy. You've done some genetics, haven't you? What happens to cells randomly when they reproduce and they double their DNA? Mutations. So mutations drive genetic diversification. When cells that reproduce accumulate random mutations, they live in an environment, right? So this environment imposes pressures. The fittest survive, and you have a clonal selection, meaning that cells with certain types of mutation expand over the others. From clonal selection, you go to clonal expansion, what I just said, and clonal expansion means genetic diversification again. And this happens over time, and then you get a multi-step branched evolutionary process in which phylogenetically lineages of different clones form by genomic alterations, and they expand according to the changing environment. So cancer as a model for genetic evolution in small. What are these selective pressures? What are we talking about? Why? What is it that is driving these clones with specific mutations to thrive and expand? Well, there could be many possibilities. You can think of many selective pressures, either from the inside of the organism or from the outside. For example, diet is something that comes from the outside, but imposes a selective pressure. Or from the inside, for example, local regulators, the availability of oxygen to that mass, the availability of nutrients such as glucose, such as glutamine, or architectural constraints. If a mass is growing and it meets a barrier, like for example a bone, then this imposes a selective pressure. Constitutive genetics is the basis on which mutations build, and the lifestyle is some selective pressure that comes from the outside. So these selective pressures continuously act on the mass as it's progressing, and it's going to change the face of the tumor. And if you take a picture of the tumor in a certain moment of its progression, and you take it a little while after, these are completely different things. Okay. So what I want you to take home is that even though a cancer is a clonal disease, meaning that it starts from a single founder cell, which is then transformed into a cancer cell, that's what we say, as transformed cell, it will genetically diversify and then develop multiple clones that may expand or shrink within the mass, so that even though this is a clonal disease, at the end of the progression, of the cancer progression, an ideal end of cancer progression, you will find that the tumor is highly heterogeneous in terms of genetics, in terms of metabolic properties, and in terms of the selective pressures that it's receiving from the environment. This is just to make a comparison with what Darwin had to do. He was working with giraffes, so you will all know the story of the antelopes that finish off the grass in the prairies, and then the ones that are born with a longer neck and that are considered ugly mates, they survive better because they start grazing on the higher trees, and then they become fittest in that environment and reproduce. This is just what happens to cancer cells. Okay, so that in the end of a cancer mass, you will have antelopes living with giraffes. The metaphor that I want you to take home. Okay, that's very, very different from what we started with. We started with only antelopes. We're finishing off with antelopes and giraffes and onyxes and all sorts of deriving herbivores. Okay, and this is a clinical example of what you will encounter when you become doctors, and some of you go into say pathological anatomy or other specialties. This is a slide from a prostate cancer in which you can directly identify by means of some diagnostic tools that I'm not going to go into details you can identify different spots in which cells have different mutations or chromosomal rearrangements or point mutations, and it's likely that each area, each region of this tumor also has different metabolic properties, because genetics and metabolism are linked processes. Genetics will dictate metabolism, and the other way around, we will have a chance to see. So, but in the end, what I want you to take home is that a cancer is a highly heterogeneous disease. And it's heterogeneity that we need to take into account when we deal with the complexity of cancer. Now, if you develop a therapy that is effective on cells that have this genetic background, it may not be as effective on cells that have this genetic background. You have to be aware of this when you go into your practice, because this is at the basis of therapy failure. And you will have to consider that when you do a therapy, it has to be as comprehensive as possible in hitting all these different cells. This is particularly true when we supply anti-metabolic therapies, which are coming up in the clinics. I'll leave you this box here. I'm going to leave you this material, if possible, and you can go and read what the implications of the Darwinian model of cancer growth brings. Okay, this is a very nice review on the topic. Some of this implication may be exploited in the therapy, in searching for new therapies, which is what we do in IH research. Some represent cancer vulnerabilities that may be hit, but some also are important because they allow to predict therapy failure, to predict why some interventions have drawbacks. For instance, why when we take out a big tumor out of a patient, you think you're doing well, but you're actually relieving a selective pressure, which means that likely metastatic spreads will expand immediately. No? So careful. It sounds like a threat, but it's not. So, of course, if there are questions or you have comments, just raise your hands, and I'm happy to take them. So, during cancer progression, as we just observed and said, heterogeneity will arise, and this will have to do, first of all, mainly to spatial constraints. Now, if you think of this as a vessel, and you think of these white cells as an initial cancer mass, what do you predict is happening to these gray cells? They start becoming hypoxic. They suffer from the distance from the vessel. Okay, so why is it not only that they become hypoxic? Why is it not just oxygen there? The problem. What else is there? If they're far from the vessel, they get oxygen. They don't get what? Glucose, nutrients in general. Let's not think that glucose is the only thing they want to take from the blood. What's going to happen to these gray cells? Guys?
Gonna start releasing inhibitors and factors.
Well, before they do that, they die. The very simple explanation. They start dying. Okay. So you get an initial shrinkage of the mass. Now I'm going to simplify this very much. Don't think it is simple, but just for the sake of it. So they start dying. Who do you think among these gray cells will survive? What property, by random mutation, must these gray cells acquire to survive?
That means less oxygen, less nutrients.
But this is going to be hard for a cancer cell. What properties do you want to develop if you live far away from downtown? Yes. So let's put it better. The gray cell among the gray cells that develops the ability to attract vessels because it produces more pro-angiogenic factors will be fittest in this environment, which is far from the vessel. You agree with me? You want to be that gray cell. You don't want to be the rest of them. If you are that gray cell Then you start attracting vessels, and you can thrive now. Now, these vessels are coming up, but they bring oxygen, they bring the glucose. What do they bring with them as well that you gray cell don't like that much? Yes. Immune system. Right. You don't want to have the immune system there, especially if the immune system. Well, as I said, I'm going to simplify. So let's keep it this way. So the immune system gets there and it starts fighting the cancer. So what's going to happen to these gray cells now? Same thing as before. They're going to die. It's a cycle of life and death. As I said, this is evolution. Evolution. There's no evolution without life and death. So which cell of these gray cells do you want to be? What property do you want to develop now? You want to attempt one?
Yeah.
Go ahead.
They want to eat from the system.
They want to hide. They want to. That's what you said. That's what you meant. They want to hide. Yes, they want to hide from the immune system. They want to mask themselves. Now, if they manage to mask themselves, now they're going to thrive because they have a couple of properties that make them really fit. They can draw vessels, so they can provide nutrients to themselves. They can escape the immune system. And so now they grow and grow and grow. And they meet a goal. They meet an obstacle. They meet a barrier. What's going to happen to them? Say it again. They die. Let's say they die. Which gray cell do you want to be? What do you want to develop now? What do you need to do to survive? They want to be able to live without being attached to other cells. Okay? They want to escape the barrier, the constraint, the physical constraint, and they want to become metastatic cells. It takes a number of properties to acquire the ability to become a metastatic cell, to leave a certain site and to move to another. But the fittest cells that are able to do this, now they travel far and they colonize. Okay? Are we done here? No escape for the patient? Is there another selective pressure that you can think of? Something that doesn't come from within, but it's given from the outside. Look at your desk. That's the selective pressure.
Yeah, maybe resistant to some.
That's the property. Tell me the pressure. It's your desk mate. That's the pressure. It's your colleague. It's the oncologist. It's the surgeon. It's the drug. That's the selective pressure. Now we're going to impose the tumor. I'm talking about therapy. So these cells now receive therapy, so we cannot ignore anymore that there's a metastatic cancer in the body. We give therapy. What gray cell do you want? Well, first of all, what happens to the gray cells?
They die.
Perfect. So which cell do you want to be?
The one.
Yes. Whatever.
The ones that are resistant to the drug.
The ones that develop resistance to therapy. The ones that develop the property of spitting out the drug as you give it. Okay. Those are the cells that thrive now and that are very likely going to kill the patient. Because we get there in the end. We get there if we don't do this on time. So there are many ways in which this evolution can select for the most malignant forms of cancer, which you will have to be aware of, because these are the multi-steps that are sort of inevitable, and that's why I'm talking about the dynamic disease. So we're talking about selective pressures, and we said that the main selective pressure, the first one that these cells encounter is hypoxia. And it's true, hypoxia is a terrific selective pressure because it pushes the cells to completely change their way of being. Without oxygen, what's the first metabolism that needs to be arrested? Halted. Now you have to go back to the first year when you did some biochemistry and you found it so boring, and so you said, why am I here seeing this, all this, you know, metabolites and stuff, what's gonna help me with in my career? Surprise me with special effects. What's the metabolism that's going to be halted, that needs oxygen? Who uses oxygen in our cells? I'm going to faint.
You have to stop the respiratory chain.
Perfect. The respiration, oxidative. I bet you all knew, but you didn't dare say it. It's the oxidative phosphorylation. It has oxygen as an acceptor. Remember this? Please tell me you remember, because I'm going to show it to you in a second and we'll make some long reasoning on it. So it has oxygen as an acceptor, and it's used to drive the proton gradients to form ATP to ATP synthase. So the mitochondria will have to stop working for some reasons, and cells will have to completely rewire their metabolism. Very important. But in the end, what we get as we move away from the vessel with a decreasing gradient of oxygen is heterogeneity, both in terms of genetics and especially in terms of metabolism, because these cells have mitochondria that are working correctly, and they do mitochondrial respiration, they turn oxygen into water, and they produce ATP, or at least they can do it, they're able to do it. These are less able to do it. These cells are just unable to do it because they suffer near anoxia over here. So very boring. I think, Stefano, you must have seen this like 1,000 times, or Erich, you must have seen this 1,000 times, and I hate to, you too, and they, oh, yeah. I mean, these guys win the prize for the most quoted and cited review ever. I wish I had written this. I could have retired by now with all the honors. These guys, they describe the hallmarks of cancer, and this is what we're going to focus on. Deregulating cellular energetics. This is one of the hallmarks this is one of the new hallmarks of cancer, according to them, not so new anymore. But it's going to be the focus of this presentation. Why do we focus on this? As I said, because cancer bioenergetic remodeling constitutes a milieu of potential therapeutic targets that we cannot ignore anymore, as anti-metabolic therapy is entering not only experimental trials, but also clinical trials as well. And you, as doctors, might have heard of the power of metformin and all that it's been said on this, which seems to be, the drug of the century, but it's not. So to start getting into the metabolism of cancer cells, we need to get to know this guy. You've met this guy before. I see somebody's nodding. Okay, so you know this guy. This is Otto Warburg, and nobody in cancer will ignore the knowledge that a cancer cell undergoes the Warburg effect. What's the Warburg effect? Since you know this guy. Well, it's written there. It's the fact that stems from Warburg's observation. By the way, he won the Nobel Prize in medicine for the discovery of cytochrome C oxidase. So this is an important guy. He observed that cancer cells in culture, with respect to normal cells, they had one metabolic property that normal cells did not have. So he was on the verge finding precisely what we were looking for earlier on, meaning that peculiarity that if I'm able to hit, then I'm only hitting cancer cells and not normal cells. And he observed that these cells produce more lactate. This was his basic observation, which made him, rise up to all the chronicles from 1930s to today. They produce more lactate, but they do so even in the presence of oxygen. So they do not suffer, or they undergo a reverse Pasteur effect. Usually when cells are in culture and they have oxygen available, they don't want to use glycolysis. Why don't they use that? Why don't you want to use oxygen? Yes? Because it's efficient in producing ATP. You'd rather produce ATP like go to the gym by making some aerobic exercise and you're not ready for it, and you want to produce massive lactic acid, feel crampy the whole week, or do you rather want to produce ATP through aerobic metabolism? You want to do the second. You want to produce ATP because it's more efficient, and with 1 molecule of glucose, how much ATP do you get? Fully oxidized glucose. Biochemistry is long, eh? Bit more than 34, 36, a bit 36, 38. Why is nobody saying odd numbers, only even numbers? Yes, 38. 38 gross, and then you have the net ATP, of course. But this observation was extremely important, because if cancer cells do not use oxygen so much to produce their ATP, and they rather do lactate, why do they do so? I'm turning this question back to you. What's the advantage of these cells? First of all, can you think of a reason why a cell would want to produce lactic acid? Huh? You don't like lactic acid when you go to the gym. Why should the cancer cell want it? In fact, it doesn't want it. Spits it out. Why can I not keep the lactic acid inside the cell? Why can I not do this? There's always a reason. Why don't you want to keep lactic acid within a cell? Because it's acidic. And what's going to do to the pH? It's going to lower it within a cell. Are enzymes going to work if the pH is lower? They're not going to work. You're going to break the cells. Okay. So why do cancer cells produce more lactate? Why do they compromise producing more lactate? Let's look at it. Let's see the reasons why. Well, first of all, I'm going to bring this quotation over here. Very important quotation by Otto Warburg itself. Warburg thought that he had discovered the cause of cancer. He wished. It was completely wrong. Never wronger than this. He said, let me quote it, word. The origin of cancer, see how modest he was. You know the story of Warburg sending the request for a grant. Did you tell them? No. So he wrote to the German office for grants for funding his own research. And instead of writing the project as he was supposed to convince the committee that his project was worth funding, he wrote only one line and he said, I need 3,000 marks. And sent it out. And they gave it to him. Because it was powerful, he had discovered the vitamin C oxidase Do you not want to fund somebody that's just asking you for 3,000 marks? You do. I wish it was that easy with I, but it's not. Thank God that there is peer review today. Thank God. So he said this: the origin of cancer lies in the anaerobic metabolic component of normal growing cells, meaning the origin of cancer lies in the glycolysis, which is more resistant to damage. The glycolytic pathway is more resistant to damage, meaning it's more resistant to mutations, but it couldn't use the mutations at the time. We're talking about 1930s. That is the respiratory component, which he knew very well. So he knew that the respiratory component, because it's composed of so many proteins, it was more subjected to damage. It was more easy for him, it was easier for him that the respiratory chain would break, then the glycolysis would be damaged, because the glycolysis is composed of a few enzymes that work in a chain. There's not very many enzymes, but there's a lot of proteins going on here, and this is more delicate. So he said, the respiratory component, damage to the organism favors this anaerobic component and therefore engenders cancer. So basically, the cause of cancer is the damage of the respiratory chain. No respiratory chain working, glycolysis is fostered, cancer. Never wronger. His observation was true that cancer cells prefer glycolysis, but it wasn't true that it was this is not working. It's not that they use glycolysis because they don't have alternatives. They choose to use glycolysis. They need to use glycolysis even though the respiratory chain is correctly working. We'll see why in a second. This is the efficient oxidative part of metabolism, 38 ATPs from one molecule of glucose. And if you only use glycolysis from one molecule of glucose, you get 2 net ATPs. Remember this vaguely from biochemistry. So you get 4 ATPs, but you're using 2, so you get 2 net ATPs. And at the end, you need to get lactic acid. It's called lactic fermentation. If the pyruvate, which is the end product of glycolysis, remember this guy pyruvate, very important. If it doesn't go to the mitochondria, but it stays in the cytosol, it will be met by what enzyme? What enzyme turns pyruvate into lactic acid? Lactic dehydrogenase, key enzyme in cancer. But then, what do I need this ATP for? I bet, I did say it because I bet you already do think that a cancer cell needs a huge amount of ATP, right? You do. You don't. You don't express yourself. I bet you do. Why does a cell need so much ATP?
Because it has a rapid cell proliferation.
Yes. To synthesize DNA only.
And DNA or nucleic acid.
You're stuck on the nucleic acids though. But if you want to replicate and you only replicate DNA, proteins, yes. What else? What's the first molecule you want to produce to make sure that you have a container? Lipids, lots of lipids. So this is called metabolism, right? The ability of cancer cells to synthesize macromolecules, which is crucial. It's crucial. How do we reach? This is what we want to reach. We don't need ATP for doing all sorts of stuff. We need macromolecules. We need lipids, we need proteins, we need nucleic acids. Because we want to make daughter cells and we want to double nearly every day. Okay, even with a lower ratio than 24 hours. How do we reach a high rate of anabolism? To make these components, by using the glucose through respiration, it's not going to help. If you want to make macromolecules, you need intermediates. If you remember the biochemistry, you will remember some textbook in which some products of the enzymes of glycolysis, as well as from the Krebs cycle, can be diverted. Remember all those arrows that you didn't explain yourself? Why are all these arrows not going in the direction? Something is going to the pentose shunt, something is going somewhere else, something is going to urine synthesis and stuff. Remember yourself looking at the textbook and saying, I'm never going to understand this. Right? You all have a glance of that. Okay. So Barber calculated in German, so I don't know if what he did was right because I never checked and I don't know German, but he calculated that when he cultured cells that were in normoxia, in the time needed by cancer cells to oxidize one glucose, 10 glucose are turned into lactate. So that's the basis of his observation. No? It's by the time the cells oxidize one glucose mitochondria, the rate of glycolysis in these cells is so high that 10 glucose are turned into lactate. They never get to the mitochondria. And this is the highest lactate production of cancer cells. When these cells were put in anoxia, so when he withdrew oxygen from cells, in the same time, well, of course, the one glucose going into the mitochondria could not go into the mitochondria. We are in anoxia now. 13 glucoses were converted in lactate. So this tells us something about the cancer cells. That they're able to produce ATP in normoxia, and they're able to produce high amounts of ATP even in anoxia. Now you see why you want to be a cancer cell with a higher glycolysis, because if you live far away from the vessel, you can still live better, live nicely and comfortably far away from a vessel. So the little mathematics, in a normal tissue, we don't have that much glycolysis going on. One glucose is oxidized, we are in a single unit of time, and you get 38 ATPs. In cancer cells, which have a higher glycolytic rate, while one glucose is oxidized, glucose are turned into lactate, so you get 58 ATPs. Now, who's making more ATP? The cancer cell or the normal cell? The cancer cell. By boosting glycolysis. Okay. The price to pay is that I produce 20 lactates. Okay, so let's see what I'm going to do with it. I'm going to turn it against normal cells. If I manage to do that, if I'm a real bastard, then I'm going to survive. And they managed to do this. In anoxia, no production of ATP from normal tissue, because the normal cells are just not ready to face anoxia. They don't have an overexpressed glycolytic enzyme array. So they just stop producing ATP. And maybe they do 2 ATP, the 2 from glycolysis. But cancer tissues, they're still able to produce 26 ATPs. So it's an advantage, and we exploit this feature of cancer cells through this, this diagnostic exam. It's the PET scan. Positron emission tomography, you're going to use it in your clinical practice. It marks the cells because they have a higher capacity of making glucose uptake. If you need to consume more glucose, then you likely need to take more from the blood. And this is what the cancer cells have developed. They have developed the ability to take up more glucose and to consume it more. And that's why they tend to take up most of the glucose from the blood, so that basically dying of cancer means die by starvation. Okay? They take away all the nutrients from the normal tissues which don't have this ability to take up so much glucose. And in fact, most patients or patients that are advanced stage cancer, they also have cachexia. I don't know if you heard about it. It's the basically the empowerment of muscles and the draining of energies. Okay, so not that I put this scheme here to confuse the opponent, because you know opponents to me, but it's just to show you how complex the metabolic picture of cancer cell is, because cancer cells need to make the macromolecules that you told me. They need to make lipids. Where are they? They need to make amino acids for proteins. They need to make nucleic acids for replicating their DNA. They're not able to do this if the glucose is completely oxidized through the mitochondria. But then they're not able to do so if no glucose goes through the mitochondria. Because as we said, and as you can see from this scheme, precursors come from glycolysis, but also come from the Krebs cycle. That's why Warburg was wrong. If you don't have a respiratory chain that's functional, there's no Krebs cycle going, there's no Krebs cycle running, so there's no metabolites that can go out of the Krebs cycle to feed the synthesis, the anabolism of these precursors. You need to have a glycolysis running fast, but you need to have some mitochondria there working as well. These are the components that you told me about, anabolism. Let's move forward. Let me tell you some of the key ones. Cancer cells cannot survive without these metabolites. The first of which being citrate. Why is citrate so important? Can you guess or can you read? Citrate is the metabolite that's at the basis of lipid synthesis. And lipids are crucial for cancer cells. With no lipids, there's no membrane, there's no organelles, there's nothing at all. There's no protein transport, there's nothing. You need lipids if you want to make daughter cells. You can replicate DNA, and that's fine, but if you have enough lipids to make a daughter cell, where are you going to put this DNA? So citrate. Now, make a huge effort, please, of locating citrate within the cell. Focus on the mitochondria, focus on the Krebs cycle. It is one of the tricarboxylic acids that's part of the Krebs cycle. Okay? It's the molecule, well, we'll picture it in a while so that it's easier for you. Glutamine. Glutamine, the most abundant amino acid in mammalians. And it's used instead of glucose when glucose is running short. Be aware of this when you decide the diet of your patients, of your cancer patients. You don't want to provide them likely, or you need to have a good balance of this glutamine. There's people that have, for years said that the diet rich in glutamine is good for cancer patients, not so much anymore. So paradigms do change as we get to learn from cancer metabolism. The reducing equivalents, NADH, NADPH. NADH is crucial because it's important for glycolysis to run, as it needs to be recycled, continuously reduced and oxidized, and also because it feeds the respiratory chain, the substrate for complex one of the respiratory chain. And we said that some respiratory chain must be working. The crossroad, at the crossroad, lactic acid or citrate. It's either of the two. Okay, for citrate, by going through acetyl-CoA and oxaloacetate in the Krebs cycle, we'll look at it in a second, or it can go to lactate, so that's crucial. So if we compare a normal cell, which will be quiescent most of the time, and a cancer cell, proliferating cells, you will notice immediately why they have a different metabolism. Well, first of all, the quiescent cell normally, as we said, oxidize glucose fully through the Krebs cycle and produces the correct amount of ATP that's needed to stay there, to thrive, not necessarily replicate. But a proliferating cell must make lipids, as we said, must make proteins, nucleotides. So it needs precursors. What happens to these cells? Follow my laser pointer, which unfortunately they don't see at home, but I'm going to read the enzyme out loud. Let's try this. So the glucose enters the cell, it's turned into pyruvate. Much of it is going to lactate dehydrogenase, but some of it is going to acetyl-CoA to bind to oxaloacetate, becoming citrate. Now, this citrate will not likely proceed in the Krebs cycle, as it normally should, to regenerate oxaloacetate, but it will be brought in the cytosol. Why? Because it's in the cytosol that you need citrate to make lipids. You make lipids in the cytosol. You don't make them in the mitochondria. In the mitochondria, you break them down through the beta oxidation. So when citrate is out in the cytosol, it's broken back into acetyl-CoA. It's like a crazy loop, but that's what cells do. Acetyl-CoA binds to cytosolic acetyl-CoA goes into lipids through the FAS enzyme, the fatty acid synthase. Oxaloacetate is generated, and then it's recycled back to pyruvate somehow. No need to go into the details. Well, what happens in the mitochondria? All this citrate is going out. What I need, what's happening here is that I have a hole in oxaloacetate. This is a cycle. To make oxaloacetate, I need citrate to go all the way around. But if it doesn't, and it's exported, oxaloacetate will not be regenerated. Now, how can I keep on making lipids if this acetyl-CoA does not encounter oxaloacetate any longer? Cancer cells must face this as their main problem. And it's the reason why cancer cells rewire, we say, their metabolism to do what we call anaplerosis. What's anaplerosis? Anaplerosis is the process by which cancer cells are able to replenish oxaloacetate. As simple as that. Simple definition. If you have a high anaplerotic flux, it means the cells are quickly replicating. Because if they're quickly replicating, it means they're doing a lot of lipids and a lot of citrate is going out of the mitochondria. Again, oxaloacetate is forming. Other reactions will replenish oxaloacetate. These reactions are specific for cancer cells. They do not occur normally in quiescent normal cells. They represent targets that can be hit in cancer cells specifically, not in normal cells. Let's see some of them. What do cancer cells use for anaplerosis? They use glutamine. They don't use glucose. The glucose is needed to form citrate that goes out. How do I replenish oxaloacetate? Through glutamine. Glutamine is a very plastic metabolite. Well, first of all, it has the advantage of being extremely abundant in cancer cells, in the organism in general, in mammalian. So as glutamine enters the cell, it can be converted by a series of reactions, which I'm not going into details, into alpha-ketoglutarate. There's a lot of reactions that can transform glutamine, an amino acid, into one of the Krebs metabolites, because alpha-ketoglutarate is a Krebs metabolite. Alpha-ketoglutarate is used to replenish oxaloacetate. So it enters the mitochondria, or it's produced by glutamine within the mitochondria, and can then move to forward into the Krebs cycle, going to succinate, fumarate, malate, oxaloacetate. If you remember the cycle, but it's over here. So we skip all this branch, we skip this part of the TCA cycle, of the Krebs cycle. Cancer cells do not have this part of the Krebs cycle working so much as they replenish oxaloacetate through gluten. Now you know why glutamine is crucial for cancer cells. By doing all this anabolism, cancer cells must produce some byproducts they want to get rid of. One of them is lactate. And we explained we don't want lactate within the cell because it lowers the pH, and this is not good for enzyme. But by doing the reactions that I told you earlier, transforming glutamine into alpha-ketoglutarate, there's a lot of other byproducts such as protons, such as ammonia. We're talking about transaminating reactions. When it comes to amino acid, the Krebs cycle metabolites do not have nitrogen on them. So alpha-ketoglutarate has no nitrogen, but glutamine does. So you need to take this nitrogen out and you produce ammonia. Do we want ammonia? No, we don't. We don't want anything that changes the pH within a cell. So protons, ammonia, lactate, they will have to be transformed by cells or thrown out of the cell to lower the extracellular pH rather than the intracellular pH. And to do this, cancer cells have an array of strategies. And please, when I say that they have an array of strategies, read this. Most of these poor cells have died. Some of them have developed the ability of expressing, for instance, MCT4, which is a transporter of lactate towards the outside. So the cells that have gained this ability through random evolution, now they are fitter to extrude the lactate. Okay? So this is the way I want you to read this. Or for example, they have acquired the ability to overexpress carbonic anhydrase to convert carbon dioxide into the carbonate. Or they overexpress LDH5, which is an isoform of lactic dehydrogenase, or the VATPase, which is a sodium proton exchange, so they get rid of protons. All these abilities, cancer cells that are fittest have acquired, and this allows them to survive even though they make these byproducts that would damage them if they kept them inside. And the lactate specifically, which is the main metabolite that is produced by cancer cells, as we just said, as Barbara taught us, I said, can be used for a number of things which make the cancer cell a real bastard cell, because it uses lactate as the rest of the environment. Now, this lactate, for example, induces angiogenesis. It's a great stimulus of angiogenesis. It attracts macrophages, and macrophages produce VEGF. Now, cancer cells, by secreting lactate, ask macrophages to do their job of attracting vessels. Diabolic, isn't it? By lowering the pH in the extracellular environment, the extracellular matrix breaks up. Now, who's going to use this property? The cells that want to move out. The cells that want to become metastatic, and forgive me if I say want, they don't think, of course, but for the sake of simplicity, the cells that are able to migrate, they encounter a matrix that is more flexible. Not only this, but imagine all this lactate going to muscles and to the liver especially. there's an ability of the liver that you might remember from clinical biochemistry. This guy here, the co-recycle. If you remember the co-recycle, the liver is the organ that is able to turn lactate into glucose through gluconeogenesis through the co-recycle. So if you are a cancer cell that's thriving in the liver, you are exploiting the liver to do co-recycle. You do all your glycolysis, all your anabolism, you put out the lactate, your cells take up the lactate, they turn it into glucose, and they give it back to you, cancer cell. This is kind of parasitism. There's all sorts of relationships between cancer cells and normal cells. So these are all the effects of lactate. I've quoted them here for you to see. And the relationships are different. Parasitism between cancer cells and normal cells, but there are models of symbiosis among cancer cells, thanks to these byproducts that are secreted by cancer cells, and which reflect specifically the metabolic heterogeneity that we're talking about. This is a vessel, and these are all cancer cells that have started from a clonal origin, but they now have differentiated in terms of metabolic properties and likely genetics, because these cells, red cells, which metabolism is these cells going to use, or which metabolisms are these cells, is this cell going to use? She has oxygen and she has glucose readily available. So if you were a cancer cell, which would you use? Glycolysis, respiration, or both? Both. Now we got the point here. I'm happy. I can go home nicely and be happy that I gave you the point. You use both. But what's the blue cell going to use? Can it use respiration? It's anoxic. It will have to use glycolysis. So which of the two is going to make more lactate? The red or the blue? The blue. Perfect. So what's it going to do with all this lactate? It's going to send it out. Now these red cells can pick up this lactate and turn it into the co-recycle if they acquire the ability to perform gluconeogenesis. Or they can use an isoform lactic dehydrogenase to turn lactate into pyruvate and then feed the Krebs cycle, which is going fast in them because we said that in both metabolites. So this cell might decide to leave out the glucose for this cell to use. How does this happen? It happens that the blue cell is selected to have a higher expression of GLUT, which is the glucose transporter, than the red cell. So the blue cell has a higher affinity for glucose, even low amounts of glucose, the blue cell is picking it up. It picks it up, it turns it into lactate, it overexpresses MCT4, which is a lactate extruder, and the lactate stays here and it's picked up by the red cell, which instead is overexpressing MCT1, which is a lactate intruder. And see how these cells become symbiotic and they can all thrive in the tumor mass. But again, they are metabolically different, which means that if you use a specific therapy that needs metabolism, you will have to consider this. This cell, the therapy might be effective on the red cells, not on the blue one, and the other way around. Now, this is a model that I just brought up from a group of colleagues in France. This is the group of Rodrigo. They usually make this very messy papers in which you lose yourself for a while, then you decide to leave it out for a couple of days and let it stay and then you take it back and then you start understanding slowly what they want to say. And you can see the mess over here, but just in case, just to simplify, they model the metabolic reprogramming over waves that correspond to gene expressions that follow the stages of cancer progression. So that at the initial stages, the wave 1, you get cell transformation. Cells become ready to proliferate. They become real cancer cells. And at this stage, the metabolic properties are only dictated by whatever oncogene, or you know what oncogenes and tumor suppressor genes are, perfect? By what oncogene and what tumor suppressor genes dictate. So every oncogene or tumor suppressor gene, you might not know this, but has huge effects on metabolism. It has been described over time. MYC, P53, for instance, we thought it was the guardian of the genome, no? We thought it only regulated cell cycle. It doesn't do that only. It regulates metabolism. It's able to boost respiratory metabolism. So when it's mutated, respiratory metabolism goes down and glycolysis is favored. So with mutated P53, you get the Barber effect, for instance. But others do other things as well. This happens during wave 1. Now, as cells start to grow, as we said, they encounter hypoxia, which is wave 2. In hypoxia, metabolism will change and gene expression will change to induce a hypoxic response that's mainly driven by this guy, HAF. Who knows HAF? Quite a number. Okay, that's good. So HAF stands for hypoxia-inducible factor. It's the main driver of the hypoxic response. The cell need to activate this guy during wave 2 specifically if they want to get out of the hypoxic status. What's HIF gonna do? It's gonna boost glycolytic enzymes and it's gonna shut down the mitochondria. No oxygen, shut the mitochondria, boost glycolysis instead. It does a lot of other stuff. For example, it transcribes vascular endothelial growth factor. If you are in a hypoxic state, you need to activate HIF if you want to draw vessels by producing VEGF. At this stage, pyruvate is just not going to the mitochondria. It's just going into lactate. As pyruvate is only going into lactate, cells need to take up more glucose because ATP production is not efficient. So they consume all glucose and you get into wave 3, glycemia. There's no glucose available, so cells need to rewire again their metabolism. AMPK will be activated here. AMPK is the sensor of energetic status of the cell. When ATP levels drop in favor of AMPK is activated. And it follows a cascade of events, changing gene expression. Mitochondria are again starting to be recovered because the little glucose that I get, I want to use it efficiently. And as I recover from hypoxia, as I recover from aglycemia, I get into mitochondrial revival. Vessels are coming up again. Now I get more oxygen, more glucose. I want to use it efficiently. I need to restore mitochondrial respiration. And I do. This is what happens through, for example, the master regulator of mitochondrial biogenesis, which is PGC1 alpha. This is an overview by the paper of Rossignol on the different roles of common oncogenes or tumor suppressor genes into metabolism. See how much these genes have an effect on metabolic rules. At a glance, you can have it there and it as a poster in your room. Now, what we learned from the role of metabolism in cancer cells came from something that I had to bring to you, because mitochondria were overlooked for a long time. focused on glycolysis as cancer cells had that pathway mostly upregulated. So we all went and tried to hit that pathway and told ourselves, if we block glycolysis, then we're going to kill cancer cells, right? This is common sense. If they use glycolysis because they need it, then if I stop glycolysis, normal cells will be able to survive through oxidative phosphorylation, and cancer cells will die. This unfortunately was never the case. There were inhibitors of lactic dehydrogenase, for instance, that made their way into experimental clinics and stuff, but they never really took up as the solution for cancer. Information on the role of metabolism in cancer came from the discovery that some enzymes of the cycle are actually tumor suppressor genes. Nobody thought before this that metabolic enzymes could be tumor suppressor genes. We thought of tumor suppressors as genes that control, and canonically that's what they teach you, at least in genetics oncology, that they are a regulator of apoptosis, or that they block the cycle so that when they're mutated and they lose their function, you get a cell to proliferate. But nobody thought that mitochondrial enzymes of Krebs cycle could be tumor suppressor genes. Now, this came from the discovery of the genetic cause of paragangliomas and chromocytomas. Now, these are kind of indolent tumors of the paraganglia, which are called pheochromocytomas. So they rise in the adrenal paraganglia, but I shouldn't tell this to medical students because you probably know. So these tumors had no genetic cause other than mutations in succinate dehydrogenase, which, as you will remember, is a Krebs cycle enzyme. You will remember this. Nearly at the same time as they were looking for genetic causes of this strange syndrome called HLRCC, which is called hereditary leiomyomatosis and type 2 papillary type renal cell carcinoma, with a lot of findings, especially from the uterus and renal cancer. The genetic cause in these tumors was nothing but a mutation in the fumarate hydratase gene, which is another enzyme of the Krebs cycle. This was strange. How come when we lose the function of these two genes, because these were loss of function mutations, we get cancer? Can you think of an answer while I speak? If I block enzymes of the Krebs cycle, why do I get cancer? That's the main hint. It doesn't have to do with cell proliferation. It doesn't have to do with cell cycle. It doesn't have to do with apoptosis. It has to do with metabolism. There's something there, metabolically speaking, that causes cancer, that transforms cells. Try to think of it. Then a third group of tumors came along, gliomas, brain tumors. Now, gliomas or brain tumors in general are very, very dangerous tumors. They have a low prognosis most of the time. But in some less aggressive forms of glioblastoma, in gliomas, they found the genetic cause was no common oncogene, was no common tumor suppressor gene, was mutations in this isocitric dehydrogenase enzyme, again, another enzyme of the Krebs cycle. And you place them here in the Krebs cycle, if you remember, fumarase, succinate dehydrogenase, isocitric dehydrogenase. Now, what's going to happen? Now, let's focus on these two, which are very nearby. They have a subsequent reaction to the Krebs cycle. Can you tell me what's going to happen if you mutate this enzyme or this enzyme? What's happening to the Krebs cycle? If you mutate succinate dehydrogenase, what's going to happen? What do you expect to happen in the cell? What do you expect to happen in the Krebs cycle? Does it go forward? What happens to succinate? There's an accumulating idea here. Who agrees with this? Not many friends in the class. Who's friend with this colleague here? Who says if I stop the activity of succinate dehydrogenase, the product of the enzyme accumulates? I vote for you. Professor Indraccolo is raising his hand, so I suggest you do the same. Erich too, of course. So succinate accumulates, and if fumarate is mutated, what is it that accumulates? Fumarate. Very similar chemical structure, succinate and fumarate. Now, to know why the accumulation of these two metabolites leads to cancer, and it's sufficient to lead to cancer, you need to know, as you told me you do, this guy here, HIF, the hypoxia-inducible factor. The metabolism of this factor tells us that this factor is only stable in the cell during hypoxia. It's called hypoxia-inducible factor because it's induced by hypoxia, meaning that it's stabilized by hypoxia. During normoxia, meaning that in all our well-oxygenated cells, this HIF-1 alpha is constantly produced and degraded because I don't want to use it. I don't need to use it. I produce it, ready to be used if I become hypoxic, but I degrade it. How do I do that? I hydroxylate this factor onto two prolines, and this mediates proteasomal degradation. Let's see better how this happens. So we have the HIF-1 alpha and its prolines. When there is oxygen available, oxygen is bound to the prolines. Not really, it goes through alpha ketoglutarate. And HIF-1 alpha gets hydroxylated and degraded. But this oxygen binds to the prolines by going through an enzyme which is called prolyl hydroxylase, which uses alpha ketoglutarate to catalyze this reaction with molecular oxygen, giving succinate as a byproduct. And this is the crucial point. Like many enzymes, the enzyme is inhibited by its own product. So prolyl hydroxylases are inhibited by succinate. Now, if I get a succinate accumulation, what's going to happen to the enzyme? It's going to be blocked. What's going to happen to HIF? Can it be degraded now? It cannot. So if HIF-1 alpha will stay stable, what did I tell you that it does? It increases the levels of which enzymes? The glycolytic enzymes. So by having HIF-1 alpha stable, I get the Warburg effect. And I also get the increased VEGF, so I get more angiogenesis. These are all properties that a cancer cell wants to have. And it seems there are properties that are sufficient for a cell to become a cancer cell.
Sorry. Unfortunately, we have to take a small break in the sense that for medical students of Medicina and Chirurgia, in theory, the lesson would be over soon.
Okay.
So I would say that if somebody has any questions,
Please ask.
please, I know you have lessons, you guys have lessons at 12:30, right? Or today you do not have lessons. Have lessons, and maybe you can continue, so maybe you can.
I was going to go for another 50 minutes, then
So
stop.
if it's fine for the students of Medicina here, it's another 41 hours, more or less, right?
50 minutes and then
Okay.
I think I've killed.
No short break.
No, but I thought it would be better, you know, since it's a bit more than we've been going on for quite some time. I realize, but it's logical to follow the discussion. Then at 12, in 15 minutes, then you're free to go.
I mean, it's fine.
So back to what we were saying, prolyl hydroxylases allow through this accumulation of success to stabilize if1 alpha constantly, and this provides the cells with the ability of constantly responding to hypoxic stimuli, even though there's no hypoxia. And this is a situation that we call pseudo-hypoxia. It happens in SDH mutated tumors, it happens in FH mutated tumors. The IDH mutated tumors is a slightly different story. I don't have time to go into details with that, but just to tell you how we found out that Krebs cycle enzymes can actually cause cancer by losing their function and becoming true tumor suppressor genes. So the same effect, the same cell transformation you obtain if you give cells large amounts of succinate and fumarate. If you give cells so much of these metabolites that are able to block the activity of prolyl hydroxylases. And that's why we call these metabolites Onco Metabolites. This is something that I really wanted to touch in this lecture because it's taking up very well lately in the literature the role of oncometabolites. These are metabolites that are able, by increasing their amount, to transform a cell, to make a normal cell become a cancer cell. Power metabolism, it's all expressed in this role of oncometabolites. Now, alpha ketoglutarate. I just told you that alpha ketoglutarate is the substrate of the enzyme. It's needed to stabilize, to degrade if1 alpha. If you have an accumulation of alpha ketoglutarate, what do you expect to happen? Do you want to be a cancer cell in which alpha ketoglutarate accumulates? No, because you're not able to respond to hypoxia. And maybe it's not a coincidence that we don't have human cancers in which we lose the function of the alpha ketoglutaric dehydrogenase. Because if we lose the function of that enzyme, alpha Kg goes up, and that's not so much compatible with cancer cells adaptation. Alpha ketoglutarate is currently considered an anti-oncometabolite. And in fact, there's a number of studies in which you provide cells with large amounts of alpha Kg analogs, and you actually decrease tumorigenic potential. But do these enzymes produce these metabolites, succinate, fumarate, and the transforming effect is only on HIF1 alpha? No, it's not that easy. These metabolites are called oncometabolites because they have a property. They bind to, they are products and inhibit prolyl hydroxylases, but these oncometabolites also bind to a class of enzymes called dioxygenases. Alpha ketoglutarate included. So as I said, caution on alpha Kg because it's currently considered an anti-oncometabolite, but it may have transforming properties in other senses because alpha Kg is a substrate of dioxygenases. Now, what is dioxygenases here? If you studied a little genetics or epigenetics, have you studied some epigenetics? You know that DNA can be modified into epigenetic terms through methylation and demethylation. These are reversible changes in epigenetics. Histones can be modified through acetylation and deacetylation, or methylated and demethylated to change the gene expression without changing the sequence of the DNA. This is the concept of epigenetics. Now, the enzymes that carry out this epigenetic modification are dioxygenases. And dioxygenases use Krebs metabolites to function. So if you have an imbalance of Krebs metabolites, you have an imbalance in gene expression at the genome-wide level. Can you imagine the damage you can do if you change gene expression at the genome-wide level because of an imbalance in Krebs cycle metabolites? You may end up overexpressing oncogenes. You may end up repressing tumor suppressor genes. So the transforming properties of these metabolites also goes through their specific action on epigenetic changes. See how from metabolism, we went back to genetics or to epigenetics in this sense. And in epigenetic changes, it's very important what happens to methyl groups. Is it not? For methylation to occur or for acetylation to occur, you need this one carbon metabolism, it's called one carbon metabolism, to run properly within a cell. And this is all metabolic. Now, monocarbonic units, as you might remember, are transported by folic acid through a cycle which includes S-adenosylmethionine. And so this is intimately connected to the Krebs cycle, and intimately connected to the amino acid synthesis pathway, which is again metabolic. So I'm not just giving you an overview of how metabolism enters into genetic expression. Acetylation uses acetyl-CoA, which is a product of glycolysis and Krebs cycle. Or at least it shifts the balance between what goes into the Krebs cycle and what goes not. So if we and by the way, let me show you here that there were drugs that were used very much in the past and still are, that we didn't know, but they are anti-metabolic drugs. We experimented them and we saw that they reduced cancer proliferation. Why do they go? Because they impact on metabolism. Methotrexate, for instance, on folic acid. This is an anti-metabolic drug. We didn't know it was anti-metabolic. We knew, but now it gains importance in the field of metabolic oncology. So definition of an oncometabolite. Any metabolite that functions as a competitive inhibitor of alpha-KG dependent dioxygenases. Most dioxygenases use alpha-KG to take that oxygen from the environment and bind it to the product. Here it is, the reaction. Product is succinate and CO2. This is the main reaction of a dioxygenase. I'm going to wrap up with the very last concept. I'm not going into detail of this again, but just to give you a full overview, because we've now talked about what happens in cancer cells, especially at the primary tumor site. But let's take a peek at the metastatic site now, because the metabolism of the primary tumor is usually different from the metabolism of the same tumor at the distal sites. And you have to keep this into consideration when you're going to clinical practice. Always consider that these two entities may be different. Now, to move to a distant site, you might know that a cell needs to learn to detach by the rest of the cells at the primary site. It has to acquire the ability to enter the bloodstream or the lymphatic stream, and then reach a distal site where it has to cancel out the properties of living detached, and it has to reacquire the properties of living attached. It's a huge amount of evolution. The process is huge. A cell has to learn, imagine an epithelial cell which is programmed to be attached and polarized, the cell has to be selected by random evolution to become partially mesenchymal, so to acquire a non-polar properties, movement properties. It has to survive into the bloodstream where it's exposed to a large amount of oxygen. Imagine the reperfusion problem. Imagine the amount of reactive oxygen species that this cell is exposed to. It has to reach a site, and by the way, it has to survive anoikis. Most important, who knows what anoikis is? You don't know what anoikis is? It's a type of cell death. You have apoptosis, you have necrosis, and you have anoikis. Anoikis is death by detachment. So when a cell is detached by other cells, it gets signals to die. This is called anoikis. Cells that are mesenchymal and they travel in the bloodstream, they have to resist anoikis. Then they reach the distal site and they have to reverse this process. So this is called epithelial to mesenchymal transition. The opposite process when the metastatic cell reaches the distal site is called the mesenchymal to epithelial transition, so the opposite. Lots of properties to make these transitions are metabolically regulated. Some of these actors, some of these players that turn an epithelial cell into a mesenchymal one are driven in fact by the HIF-1 factor. So HIF-1, imagine this is common sense. If 1 alpha reads out hypoxia, it means there's overcrowding. It allows the cell to acquire properties for the cell to detach. It makes sense, doesn't it? Evolutionarily speaking. And other actors as well, but I don't have time to go into details. But just to show you a quick peek of why primary tumors are different metabolically speaking from metastases. For example, in brain tumors, in brain metastases acquire different properties from the primary tumor in which they rewire their metabolism. So, for example, that they start feeding on branched chain acids because they are available in the brain much more than elsewhere. Or because the brain is able to use ketone bodies, remember this, metastases in the brain acquire, different from the primary tumor, the ability to feed on acetate. So they overexpress this enzyme, acetyl-CoA synthetic enzyme 2. And in this way, they're able to thrive in the brain, not better than they were to do so in the primary site. Metastasis in the lungs. These reach the lungs, and the first obstacle that they encounter is that there's a lot of oxygen. This is a very oxygenated place. So they would die by oxygen reperfusion because it would boost their production of oxygen species. So these cells adapt there by overexpressing enzymes such as peroxyredoxin 2 or other enzymes of the redox system, so that they're able to toxify all these reactive oxygen species. They upregulate PGC1 alpha, which is a master regulator of mitochondrial biogenesis. They increase the number of mitochondria. If there's more oxygen, then I might as well exploit it. So lots of mitochondria, so that I can also turn this oxygen into something inert like water, because I don't want to have all this oxygen, which tends to make radicals. The upregulation of pyruvate carboxylase to avoid oxphos damage, kind of complex now to go into details about. Liver metastasis, we talked about this. These metastases here are able to use the core recycle of the nearby cells to do gluconeogenesis, or the bastards, they overexpress and secrete the creatine kinase. So the creatine kinase in the microenvironment phosphorylates creatine, then this is re-imported into cancer cells, and they make ATP out on the expenses of the liver cells. Bone metastases. Well, in the bone, they overexpress osteopontin, the metastases, so they can mask themselves from the immune system. This is the problem in the bone. The bone remodeling requires a huge amount of macrophages being there all the time for bone remodeling. You want to hide from them, so they make all sorts of different masquerades, and as well produce a lot of lactate to mimic and feed osteoclasts over there. And these are called osteolytic metastases rather than osteoblastic. There can be of two types. This material I can leave to you. Last, ovarian cancer, something that both I and Stefano are very concerned about in our research lines. Now, ovarian cancer tends to metastasize at the omentum, which is this lipid-rich environment. And cells there, and they need to adapt, and they do so by upregulating fatty acid binding protein 4. They force the adipocytes to secrete fatty acids, they pick them up, and then they break them down into mitochondria, where they have a higher rate of beta oxidation, and they make nutrients out of that. So lots of strategies. Omental metastases have a very different metabolism from ovarian cancer primary site. Last slide, and with this, I let you go. Why did we say all this? Why did I give you all this overview of metabolism? Because therapies are now being tried hitting specific sites of metabolism. Something that is that I'm very fond of because I work on these things is the ETC inhibitors, the inhibitors of the respiratory chain. And metformin appears to be one of those. Now, metformin, as you might remember, is an anti-diabetic drug. It's needed to lower glycemia and glucose levels, of course, in the blood. And its action, it's inhibiting the complex one of the respiratory chain. Now, we believed until a little while ago that if we block the glycolysis, we would kill cancer cells. Well, we try to kill them now by blocking the respiratory chain. See how funny it gets. Because when you inhibit the respiratory chain, cells are not able to respond to hypoxia anymore. Because metabolites of the Krebs cycle that are slowed down are not able to, are actually more able to feed the reaction of the prolyl hydroxylase. Very long story, very complex. Lots of other inhibitors today, they act specific on enzymes or on proteins such as transporters that are crucial for a cancer cell to carry out its metabolic activity. For example, inhibitors of the lactate transporters are being tried today, or of the glutamine synthase. We said how important it is for a cancer cell to have glutamine available. I guess this is it. A very broad overview, somewhat superficial, and you will forgive me, but this is all I could do in 1 hour and a half. I usually have a course on cancer metabolism that lasts for 4 hours at the students in Bologna. Yes, it's a monographic course for the students of medicine. Of course, we go into details, and there's a lot of evidence involved, so you wouldn't like it. But and other than no, actually they do like it, but they are first year, so they have the biochemistry fresh at hand. So thank you very much for your attention and questions.
I have a question to start with. So do you think metabolic therapy would be sufficient to kill cancer? Or have to be associated in combination with classical cytotoxic therapy, for example?
I'm not sure of what would be most efficient to associate it with, but definitely not anti-metabolic therapy alone. And this is already proving to be a drawback we use only anti-metabolic therapy, cancer cells have such a plastic ability to rewire their metabolism. Giving only one compound to block one of these metabolic routes is not going to be sufficient. In our research line, we already identified adaptive pathways that arise when inhibitors are given. If you give an ETC inhibitor, you can have a rewiring of metabolism towards glutamine carboxylation or reductive carboxylation of glutamine, for instance. You're going to block HIF1 alpha, but then there are non-cell autonomous mechanisms that ensue. So I'm not sure if we have never tried the combination between a common cytotoxic or cytotoxic therapy plus an anti-metabolic therapy, but maybe combining more than one metabolic therapy could be a way. I guess it's too soon to say.
And one last thing. Are there many side effects to these? So cytotoxic class chemotherapy, one knows what sort of side effects one has. If we're targeting these metabolic pathways, are they much less toxic than the...
They seem to be much less toxic because they go and hit metabolisms that are either differentially carried out or differentially expressed by cancer cells. Again, the story I know best, of course, is again the complex 1 inhibitors. Normal cells can survive being exposed to complex 1 inhibitors because cancer cells use it much less, so you can play on the doses there. So it becomes a little more effective on cancer cells than in normal cells. I foresee less toxicity if we manage to hit the correct metabolic pathway, or in the case of tumors where the mitochondrial enzymes are mutated, drugs that are specifically targeted on the mutated enzyme. And that's the easiest possibility.
From you guys, any questions?
They're dead. Like
There's
one question.
I'm survivor.
I'm thinking, with this kind of speed and ability to adapt to anything, basically, like, I don't know, it makes the point of trying to target metabolism when they can adapt to many, many different conditions that normal cells will never be able to live in.
Well, again,
they're gonna adapt anyways, in a way.
No, I don't agree with that. So such a pessimistic approach.
It's a pessimistic approach.
It's a pessimistic approach. It's worth trying because if you manage, as we were saying, with the common therapy to hit more than one crucial pathway, which is still specific to metabolic, to metabolism in cancer cells, then I think that's the way. You want to do this because, first of all, you want, again, going back to the very beginning of the lecture, you want something that's specific for cancer cells. You want to give a therapy to the patient that avoids heavy collateral side effects. And this is a way to do that. At least our understanding that metabolic routes are differently active in cancer cells provide us with a choice rather than basing therapy on cells that are simply divided, because there's a lot of cells in the body that divide at any time, and on the long run, cytotoxic therapy can actually cause cancer rather than, and we all know this. So I think the combo therapy is a way, and there will be ways in which the metabolism is so cut out from different ways that you don't have any more choice to produce, to do anabolism as it's needed.
So giving the toxic drug, you also, you said that you also DNA reduction, so you also inhibited the reprogramming in a way.
Possibly.
Any questions from you guys connected via Zoom?
But optimism is the first thing. Don't forget. Let's not forget optimism.
Questions from the chat?
everything has been super clear.
clear.
Super clear.
Yes, I believe so.
Thank you again.
Thank you for attending and for your patience.
And that was also our lesson. Medicina and Chirurgia course is a bit late. We are a bit in advance, but if you agree, we can stop. Catch back the remaining minutes next week.
Okay.
Is that okay? Okay. Thank you so much.
Okay, thank you.